Well, hello everybody. This is Dr. Neary back again with a short video about the second exercise in lab number 5. And that's a photosynthesis exercise. Specifically, this is a floating disk assay looking for photosynthesis. So this formula at the top of the board is the net formula for photosynthesis. So we need 6 CO2 plus 12 H2O plus the energy from sunlight gives us C6H12O6, which is glucose, plus 6H2O plus 6O2. So we're going to be using a floating leaf disc assay using a type of plant called English ivy. So normally leaves will float on water. You've probably all seen this. The reason for that is there's air trapped inside of a leaf. If we look at the structure of a leaf, so there's, imagine this is a cross-section of a leaf. So we have one layer of cells on the outside, another layer of cells on the lower side. In the middle of the leaf, there's a loose arrangement of cells called mesophyll. And also there are some openings in the bottom side of the leaf called stomata. These stomata are important because they allow air into the mesophyll of the leaf. Air is important because that is the source of CO2 for photosynthesis. So we need CO2 in order to do photosynthesis. Now in this floating leaf disc assay, we're going to evacuate the air out of the leaf discs so that they no longer float, but rather they sink. Okay, so we do that by putting just a little bit of dish soap inside of a solution of water. This breaks the surface tension so that water can get into the small spaces inside of the leaf, and then the leaf disc is going to sink. Okay. One problem, once we do that, air can no longer get into these little spaces in the leaf and can't take CO2 inside the leaf where photosynthesis happens. We can get around that though. If we add sodium bicarbonate, NaHCO3 and sodium bicarbonate, this adds a source of carbon dioxide to the water so that photosynthesis can happen. All right, so we're going to look at a couple different conditions. We're going to infiltrate our leaf discs in a syringe so that they sink to the bottom, and we're going to look at a couple different conditions. We're going to look at a condition where we have a source of CO2 from sodium bicarbonate, but no sunlight. So think about what might happen if we have CO2 but no sunlight. We're also going to do an experiment where we have no sunlight but CO2, and we're going to do an experiment where we have all the conditions for photosynthesis. We have a source of CO2 plus sunlight. Now, if we meet all the conditions for photosynthesis, we should get this reaction. This reaction produces O2, gas, oxygen gas, right? So if we get photosynthesis, oxygen, gaseous oxygen, should begin to accumulate in the inside of our leaf discs. Oxygen gas floats. So if we start accumulating oxygen, the leaf discs will float. That means we have photosynthesis. Okay? So think about what the results might look like. If we do this experiment, this floating leaf disc experiment, in the presence of light, but with no sodium bicarbonate as a source of carbon dioxide, is photosynthesis going to happen? If we have the sodium bicarbonate as a source of CO2, but no sunlight, is photosynthesis going to happen? And are we going to get floating leaf discs? So, that's about all I have to say for exercise number 2 of your of lab 5 today. I'll be back shortly with another short video talking about the final exercise for today, where we're going to be looking at aerobic respiration.